if you're clear in an electron transport chain now, and we have completed glycolysis, Krebs cycle, and electron transport chain, we are done with the whole amount of ATP that's formed in form of substrate level phosphorylation or by oxidative phosphorylation in terms of chemoosmosis. But now we are going to count the total amount of ATP that's formed during the process of respiration from one molecule of glucose. I'm going to make this total so that you can understand and we can count the total amount of ATP. Say for example, during glycolysis, we have produced 2 molecules of ATP. We actually produce 4 molecules of ATP by substrate level phosphorylation, but we consume 2 molecules of ATP. Therefore, net 2 ATP are produced from glycolysis. And we have also produced 2 molecules of NADH during glycolysis that will also oxidize during electron transport chain. But actually, in our textbooks, we study NADH actually enter inside mitochondria. Therefore, it gives electrons to FADH2. And when FADH2 is oxidized, it actually always produces 2 molecules of ATP. Therefore, NADH synthesized during glycolysis, when will oxidize during electron transport chain, they will produce not 6 molecules, but they will produce 4 molecules of ATP. Therefore, we are reducing 2 molecules of ATP as we study NADH synthesized during glycolysis can't enter inside. They give electrons to FADH2, and when FADH2 oxidizes, it always produces, each FADH produces 2 molecules of ATP. Therefore, the NADH of the glycolysis always produce 4 molecules of ATP. So the total ATP output from glycolysis is actually 2 substrate and 4 from electron transport chain. And then we studied acetyl coenzyme. Acetyl coenzyme produces 2 molecules of NADH, and when they oxidize during this chain, electron transport chain, they actually produce 6 molecules of ATP. They are producing 6 molecules of ATP. And then finally, Krebs cycle. And Krebs cycle actually produces 2 molecules of ATP on a substrate level, and it actually produces 6 molecules of NADH, which finally, when oxidized during electron transport chain, produce 18 molecules of ATP. And Krebs cycle also produces 2 molecules of FADH2 also. And when FADH2 oxidized during this process, they produce 4 molecules of ATP. And if we count all this ATP, that will become total 36 molecules of ATP that are produced during a respiration taking place in a 1 molecule of glucose. This is a total count, and this is a 36 is a net ATP output from 1 molecule of glucose. And if we can include all the ATP during glycolysis, if we consider 4, then there would be total gross ATP output would be 38 molecules of ATP, and out of which we consume 2 molecules during glycolysis investment phase, and again we will have a net molecules 36 molecules of ATP from a respiration in 1 molecule of a glucose in a eukaryotic cell. But on the contrary, in a prokaryotic cell, as we say, this is all taking place in a cytoplasm and mitochondria is absent, and therefore we say there is no conversion of NADH into FADH formed during glycolysis. Therefore, these NADH, especially in a prokaryotic cell, produce not only 4, but they produce 6 molecules of ATP. And the rest of process remains the same, but as there is a difference of 2 ATP molecules, therefore in a prokaryotic cell, we always produce 38 molecules of ATP. This is a total count of ATP from a 1 molecule of glucose, and now we are done with respiration, and we are almost done with the most important topics of bioenergetics. Subscribe my channel, share it with your friends also, and comment as well, like as well, and let's learn together. Thank you very much.